interactions between small molecules and biological macromolecules and drug design. And before I hand it over, I wanted to hand it off to our host today, Tigis, just to give an intro in case you haven't been here before.
Hey, my name's Yara. I'm a machine learning engineer here at Tigis. Welcome, pizza, drinks, whatever you need. You let me know if you want to know where the bathroom is. So just a little bit about Tigis. One, I'm new. I've been here like 3 months, so don't fault me for anything I say. But we are a financial research company that provide data for analysts and, hedge funds, sometimes VCs, sometimes PEs, and we provide expert transcripts as well as BAMSEC filings that they go onto our platform, and they just find all this information in one place. We also provide them with some machine learning. We have an assistant. We have things like that. So yeah, if you have any questions about Tigis or even the financial data space, let me know. But yeah, other than that, please enjoy the talk.
All right, thank you for the invitation. So I'm going to talk a bit more on the biochemistry side than data science, although all the work that's been done was done with Python packages like matplotlib and NumPyro for the Bayesian regression. Well, there's also PyMC that we used. So these are all, this is all done with open source packages, and we released the code for these as an open source package as well. Well, we released the source code. I wouldn't necessarily call it a package. It's just available to use and repeat these kind of measurements if you have the data in place. So what I want to talk about is our efforts to use biochemical assays to predict cellular assays. So the biochemical assays are pretty cheap in the scheme of things. Like you can have something in a plate in the open air, and you just need to make a protein. And that's a way you can test, or a first way to test whether a drug is a good, or drug lead has a possibility of becoming a drug. But ultimately, we don't want to test drugs in a petri dish, or we don't want to use drugs in a petri dish. We want to take drugs, or people want to take drugs. And there's a large number of steps to go from a chemical that someone is trying to start out with and a drug that someone could take in the clinic. And this research that we were engaged in is just a small part of this long process. And this small part is linking something from a protein target to a cellular target, whether it could actually get rid of the virus growing in cells and allowing the cells to grow. This research was done pretty much all by one PhD student who just defended her thesis and moved to California, and so I'm here instead of her. And she was also advised by Lulu Kang, who was an applied math professor at IIT, who helped us, or just made sure that we were on the right track with the statistical methods and so forth. She's subsequently moved to University of Massachusetts, Amherst. And I just want to also briefly introduce my other members, or that were my research group early in the year, and the general areas of research in my group. So we work on computer methods to study the interactions between proteins and small molecules. You probably know that you eat protein, but you're also, protein is a big part of your body, and pretty much, or a large fraction of drug targets are proteins. And when I talk about ligand, I'm talking about small organic molecules that interact with proteins in specific ways. And pretty much all of drugs, or a large fraction of drugs, are small molecules that bind to proteins and either stop them, they turn on a signal, they turn off a signal, they stop them from doing catalysis, from making a reaction faster. And what my group has been working on are ways to use computer simulations to predict how tightly a ligand binds to a protein and whether it's going to be possibly a suitable drug. So you want something that binds tightly to one protein or a small number of proteins, opposed to if you put it in the body, it will bind to everything and cause all these crazy side effects. So most of the time, when people or when the pharma industry produces a drug, it has a very strong affinity for one protein. What my group has also worked out are ways to say, after you have the small molecule bind to the protein, how does it change the shape of the protein, and how does the shape of the protein affect the signals that it passes along? So about a third of drugs in the market target a class of proteins called the G protein coupled receptors, which are on the surface of cells. And most of the time, it's not clear exactly, well, so these GPCRs are signaling proteins. You can bind a chemical and it just passes a message through the GPCR and into the cell along multiple pathways. But it was unclear, or it has been unclear, how you actually pass a message along one pathway or another. And my group has worked out ways to go from simulations of the ligand bound to the GPCR to work out which signals are passed along and by how strongly. And that was the basis of a company that we just started. But I won't talk about that in great detail today. We also have been using deep learning. This deep learning methodology has been based on JAX code that we wrote. It's all on GitHub. And so we work on the methods, but we also have some applications to trying to understand the behavior of small molecules and proteins, trying to discover antiviral drugs, antibiotics. And the area that I'm going to talk about today is the statistical analyses of different experiments using code that was written from Python packages that are open source. Okay, so I want to briefly, everybody knows, even if they're not biologists, chemists, or scientists, everybody knows about how dangerous the coronaviruses are. Everybody knows about SARS-CoV-2. You may not know about the Middle Eastern Respiratory Syndrome coronavirus, which if you get it, you're much worse off, or your chances are not as good compared to the SARS-CoV-2, which was much more widespread. And because of these recurring coronaviruses, there's been interest in designing drugs that will work against not just one coronavirus, but multiple coronaviruses. And that's a goal of one of the efforts I'm part of, one of the consortiums I'm part of, is to target the SARS-CoV-2 or the coronavirus main protease. So this is a protein that's plays a really important in the life cycle of the virus. The virus has to go into a cell, and it causes a cell to produce what's known as a polyprotein. So this is basically like if you're trying to make a book, if you're trying to print a book, it's just like if you printed one long sheet of paper, one long papyrus sheet or so. And what the protease does is sort of like if you cut that piece into different lengths or and then you could, or cut into different smaller sheets so you can bind it together into a book. Well, that's analogous to what the main protease does to the polyprotein to make all these parts for the virus so that it can reproduce. Well, that's what the virus wants to do, but we don't want it to do that. We want to stop it. so that the body could take care of the virus. And the small part that my group was involved with is trying to connect something that happens in very mild conditions in a biochemical assay. Like you just make a protein and you can test its activity, and you can see if you add a drug to it or a potential drug, whether it actually works in stopping the activity of that protein. So that's a biochemical assay. And the cellular assay, which requires much higher safety standards and it's just a very expensive and difficult experiment comparatively, because you're using live virus and you don't want to infect your technicians who are working on it. So it's much better if we can filter all the possible compounds with a biochemical assay before we do a cellular assay. So the way that this is done, or usually done in a biochemical assay, is on a large plate where you have different conditions in different parts of the plate, and you usually change how much of the ligand you put in, the concentration of it. And usually the concentration will change the activity. So if you don't have any of your inhibitor, you're going to have 100% of your original activity. And as you increase the amount of inhibitor, then you reduce the activity. That's just what this curve looks like. And you can fit a function to this curve. It has a maximum, it has a bottom, it has a concentration where you have 50% of the activity. This is known as the IC50, and that's a key parameter that comes out of the fitting to the curve. There's also a coefficient that describes the slope of this line right here. That's a normal concentration response curve. What people have observed with MERS coronavirus biochemical assays is this, and that's why this consortium got into contact with me, because they didn't know what to do with data that looked like this. Because usually you add a little bit of inhibitor, it causes the enzyme to stop working as well. But what happens here in these data is you can add a little bit of inhibitor and it will actually cause the enzyme activity to increase. You're actually doing the opposite of what you want to do. You don't stop it, you actually make it stronger. And then if you added more and more inhibitor, then you get a inhibition as well. So they call this the activation phase and inhibition phase, and they call this a biphasic curve. But when you have this biphasic curve, it hasn't been clear, what does this mean? Does this mean that, or what do you look at in this curve to decide whether you have a good drug or not? Because if you have a standard curve, you can just say, okay, here's the IC50, the middle of this curve. But if you have this crazy biphasic curve, what do you even try to do? So some of the simple attempts to analyze these data that were actually first initially tried by the consortium are what we call the inhibition PIC50 and the control IC50. So the inhibition PIC50 looks at the inhibition phase of the curve and looks at the 50% point of that, just kind of ignoring this data. You can also say, let's normalize it or force 100% to be at the control value. So then it's a normalizer, or it's a different point. It's a bit of a shift compared to the inhibition PIC50. So we had a hypothesis is that we can predict what happens in the cell based on a full model that describes what's going on in the biphasic curve. And we built this model, so we built this in Python, and in biochemistry textbooks, you'll see models where you can find an analytical solution that you can say X equals something times something or some polynomial. And that's nice and that's helpful to have, but we don't actually need to have an analytical solution to this. We used a numerical solver to find a solution to a system of equations that is specified by this diagram. I guess I'll just explain this diagram a little bit in that there's different possible species if you have a solution that contains your protease and it contains a substrate. So that's the stuff that the protease processes and breaks up to make products. You can also have an inhibitor. That's the red stop sign. So we can say we have a single half of a protease enzyme not doing anything, and that could just be floating around in your beaker or your petri dish. Or you can have two of these monomers come together and form a dimer. And beyond this simple equilibria, beyond this simple going back and forth between these two forms, you can also have the addition of a substrate. So then it sticks to the site, to the catalytic site, and the substrate can break up into products and form a monomer again, for example. Or you can bind to an inhibitor. You can also have the dimer bind to an inhibitor. You can have the dimer bind to two inhibitors because there's two possible places for the inhibitor to stick. So the assumption in this model, or that we made in this model, is that there's an equilibrium between all these states here that occurs fast compared to the rate of catalysis. And this is a pretty common assumption in biochemistry, but they often are typically using simpler models. But we decided that we needed to have this more complex model in order to describe what was going on. So here's all the equations behind the model. So these, if you remember from chemistry, are, well, chemical equilibrium equations where you have an equilibrium constant and you have concentrations of different species, or ratios of concentrations of species. These are all chemical equilibria, and these clarify there's loops in the model. And these are conservation of mass, meaning that like if you put a protein in there, this much protein in there, it has to be in different forms. You can't lose, or you're not destroying or creating protein or substrate or inhibitor. And the rate of the reaction is just a sum of some constant times the concentration of different species, where the species is bound, or the substrate is bound to the protein in different forms. So what we hypothesized is that if we fit our data to this more complicated model, we can predict what happens when you have a model that's less complicated, when you have only the dimer. Because the literature or other people had suggested that the dimer is the only active form of the enzyme. And they also suggested the biphasic curve comes from the dimer formation when you add a small amount. That's what causes the activation to occur. So we said, if we don't have the monomer around, we won't have the biphasic curve. We'll just have an inhibition phase. We can predict the inhibition phase and get the IC50 of that phase and say this is what's going to happen in the cell. So we built a model and we did a Bayesian regression. So I heard before the talk people talking smack about Bayesian regression. Or I heard secondhand that this is useless. I'll tell you that it could be useful. And you'll see, well, the reason it's useful is because you can make models very complex. And you can overfit them with a nonlinear regression. Or you can overfit any data, right? You have data, you can make a more and more complex model. You'll find some way to fit it. Doesn't mean it makes any sense, or it doesn't mean the model is any good. But if you're doing a Bayesian regression and you'll find out whether you have like a multimodal posterior. You'll know if your posterior is really broad, that your data is really telling you anything about that parameter. You'll also be able to incorporate priors information. So there's a lot of benefits to this approach. And yeah, first thing we did was a nonlinear regression, right? But we were concerned about overfitting, so then we started working out a Bayesian model. Then we also thought, okay, still, this is a complex model. Can we make it simpler? And it turned out in this particular case, if we made this simpler, the fit just got bad, so that we knew that we had to use a more complex model. Okay, so first problem is if there's not a lot of data or a lot of different types of data, and we have a really complex model, then we can fit anything to that data. But we had this paper which had described many different experiments. And here's just a subset of the experiments. Different types of biophysical experiments, looking at how much monomer there is, how much dimer there is when you have different conditions. Looking at the rate of the reaction, a kinetics experiment. And this looks fine. Looks good. Good starting point. But this is a nonlinear regression. You try to get error bars from nonlinear regression, they're always tiny, or usually. They're all very, very often tiny and unrealistic. So we got this, but we didn't really believe it. So, okay, I already defended Bayesian regression without saying what it is, but well, the general idea is you have Bayes' theorem, which says that the probability of parameters given data is so that's the posterior is the prior. This is the probability of parameters before you do any experiments. So you know some values of the parameter is impossible, or there's another experiment by someone else. You can just roughly say that this is the area of this parameter. And this is a likelihood. What is the probability of observing the data if you had that particular parameter? And the likelihood could just be assuming that you have, for example, we typically just assume there's Gaussian noise. Oh yeah, and this is a proportionality. This doesn't really matter, but so we just know the proportionality of a probability of a certain parameter set. And we sample, we do Monte Carlo sampling from this. Okay, and this is the source of our data, our initial source of data, this paper. They had 6 different curves. They had 6 different models. They fit to these or something like that. They didn't fit a model to this biphasic curve. So we just wanted to use this as a test bed. Like, can we do a global fit to all these curves with the same model, same parameter set? And yeah, most of the parameters that we used were basically straight from this model. But there's also some nuisance parameters relating to like the noise levels and so forth. And we used the Bayesian regression tools within Python packages. I think it was PyMC4, if I'm remembering correctly. Okay, so then we get results. We want to check that our posteriors are converged. So we want to make sure that we don't get big changes on the posterior if we do more sampling. And that's pretty much what this figure shows. We also want to confirm that the most maximum at posterior, the most probable, or the one with the highest posterior density is actually fits the data well, and that does look like it's a case. And then we could start looking at what are the what are our credible intervals or our high density intervals? What are the values that the parameter is likely to be between? And in some cases, we see that it's pretty narrow, some cases pretty broad. We can get a pretty good idea of how much the data tells us about these parameters. But some of these parameters, like the rate of each individual species, the data didn't tell us very much. So we would need new experiments or different types of experiments to know about these parameters in particular. And there's also some correlations between some of the parameters that the data fit well either by having log k IDI high and log k ID low, or inversely proportional in that particular case. Okay, so that demonstrated to us, or had a good idea that we could actually fit this model to data and get some idea of how tightly the parameters are known. But that was only for one fairly small data set. We wanted to see whether we could apply this to multiple inhibitors. Because in a drug discovery campaign, they'll make hundreds of variations of a possible drug before they actually decide on something to test in the clinic. And the consortium we're part of is focusing on the Middle Eastern respiratory syndrome. And they collected these data upon our request, where they changed the concentration of the enzyme or the substrate or the inhibitor. And these data on the left are only with an enzyme in different concentration or different concentrations of enzyme or substrate. Or on the right, they also have an inhibitor, or one of the many inhibitors. So we decided to fit a subset of our model to the enzyme and substrate data, so we don't worry about any of the parameters related to inhibitor binding. And then subsequently, we considered incorporating the inhibitor binding. And yeah, these are very similar methods as we have done before with the Bayesian regression. And we have results where we can fit curves or data where you have multiple enzyme and multiple substrate concentrations and multiple inhibitor concentrations. And we did this for many different inhibitors. And we do this all at once, or we fit these all at once. So these are for different inhibitors. Some of the parameters in this are shared. Some of the parameters are different between, because you have different affinity, or each inhibitor has a different ability to bind to the protein. But then some things about the protein are the same. So those parameters related to the protein are the same in this global fit. We can also see that the credible intervals, the highest density intervals, are smaller. Like we have a better idea of what some of these parameters are when we have more data. Okay, then we were pretty happy with our progress. It took a while because we had to figure out how to incorporate some nuisance parameters or some ways to make the fitting better. But ultimately, we got to the point where we said, okay, let's try to predict what's happening in the cell and see whether there's a correlation between our predictions and the cellular results. As a reminder, we just sort of ignored anything in our model that had to do with only the monomer, and we looked at parts that had to do with the dimer. So we did our to the biphasic curve with the full model. We predicted the behavior if you only had the dimer, and then we analyzed this curve and then looked at its correlation with the cellular data that had been collected up to the point. And this is what we see. So here are a bunch of different correlations. from different biochemical assays and cellular assays. So as I mentioned in the beginning, you have the control PIC50 and the inhibition PIC50 are based on just fitting half of the data in basically the inhibition phase. You also have the dimer only PIC50 here, which is our prediction, and the cellular PEC50, which is the more expensive experiment that's more dangerous and has to be done super carefully. And we can see there's a lot of correlation between like the control and inhibition PIC50. And actually, a lot of these biochemical are correlated with each other. And they're also all kind of correlated with the cellular PEC50. So this was a demonstration that this biochemical, at least for these chemicals, the biochemical assay were providing predictive value to determine whether compounds should be advanced to the cell assays. But what we saw is that of all these correlation, all these correlation metrics show that this, sorry, I can't see all the titles of this thing here. Let's see. Yeah, the inhibition PIC50 has a similar Pearson R compared to the dimer only PIC50. So we didn't actually gain a ton with Pearson R, but we did get an improvement in the Spearman rho and Kendall tau, which are rank correlation metrics. We also looked at, oh yeah, this is the root mean square deviation between the metrics. And that's also improved with the dimer only PIC50. The ARMSD is if there's a correction so that the averages are the same. We also had an idea that we should look at not where the midpoint is, but we should look at 90%. Like it may take, or it may require you to block a large fraction of the enzyme in order for there to have any effect on whether the cell can recover from the virus. And we wanted to see whether that was better correlated than the IC50s. And we did see that's the case, and we did see that the dimer-only PIC90 is a better prediction than the IC50, according to the Pearson R and the Spearman rho and Kendall tau. And also, well, on the other hand, the root mean squared deviation was slightly higher, although when it's considered or mean corrected, it wasn't higher. So just want to start to wrap up that basically we showed that if we fit our full model to the data and made a prediction of what would happen, it would give us a better picture of what happens in the cell. And it was better than these simple metrics that had been tried by the consortium prior to our work. So this means that we could use, or the consortium can use this data analysis of the biphasic curves in early stages of drug design before advancing a subset to the cellular assays that in later stages. And this could be used beyond this particular consortium by other groups that are trying to find inhibitors of MERS coronavirus or any other protein really that has a biphasic curve in which the physical justification for that matches our model. So I want to thank the authors who provided the data that we analyzed, and also the consortium that provided their data to us, as well as collected some data to analyze, and you for your attention and joining in this talk. Questions? Oh, so